Alpha-1 antitrypsin deficiency, or AATD for short, is caused by mutations at the SERPINA1 locus, the gene that codes for alpha-1 antitrypsin, or AAT. AAT is a crucial protease inhibitor and most active in the lung tissue to protect alveoli from elastase activity. The most common mutation is the Z mutation, and people homozygous at this locus have 10 to 15% the normal levels of AAT in the blood. Inadequate levels create an imbalance between proteases and inhibitors, leading to degradation of lung tissue, causing emphysema and chronic obstructive pulmonary disease. The deficiency doesn't result from AAT not being made, but rather is due to a tendency of Z-type AAT to polymerize with other molecules, accumulating in the rough endoplasmic reticulum of cells. This accumulation prevents most AAT from being secreted into the blood, leading to the aforementioned lung problems. 80 to 90% of AAT is produced in liver tissue, and this accumulation greatly damages hepatocytes, often leading to cell death. This causes scarring and fibrosis of the liver, which greatly impacts the liver's crucial functions in the body. Lung damage can be stopped by infusion of AAT directly into patients from donor plasma. However, there is no effective treatment for the liver damage from Z-AAT accumulation until the liver is so damaged that it fails, and a transplant is necessary.
To address this problem, researchers focus on finding a treatment for the buildup of Z-variant human alpha-1 antitrypsin, or Z-HAT, before the accumulation leads to significant liver damage where a transplant is necessary. A recent study explored an avenue to do this by disrupting the polymerization of Z-HAT through small molecule inhibition. Using a molecular docking program, candidate compounds most likely to provide steric hindrance and disrupt polymerization were identified. Specifically, those that targeted the chain of residues that constitute the polymerization interface of Z-HAT were selected. These candidates were tested in both in vitro hepatocyte cultures from transgenic mice as well as in vivo transgenic mice. UFC1 proved to be an effective candidate both in vitro and in vivo. An overlapping signal from biotin tagging in vitro confirmed binding of UFC1 to Z-HAT. And more importantly, the signal was not localized to the endoplasmic reticulum, where aggregation of Z-HAT usually occurs. This suggests successful binding of UFC1 to the polymerization interface and disruption of proteotoxic Z-HAT aggregation.
In more quantitative findings, the hepatocyte cell cultures exhibited lower intracellular levels of Z-HAT. Additionally, the presence of UFC1 did not affect albumin protein levels in Z-HAT cells, nor did it affect wild-type HAT levels. In vivo results from 3 months of treatment in transgenic mice provided more evidence. Both immunochemistry and Western blot analysis exhibited decreased intracellular Z-HAT levels in the liver, along with the same lack of interference with other proteins as the in vitro hepatocytes. These findings suggest that UFC1 prevented polymerization and accumulation of Z-HAT with high specificity for targeting Z-HAT proteins.
This study showed that small molecules can be selected to bind and stabilize the structure of AAT by analysis of previously solved crystal structures. The results of their study suggested that the small molecule UFC1 may inhibit an alternative process of Z-HAT polymerization, as both intracellular and extracellular Z-HAT levels were decreased in the cell model. The results also suggest that the interaction between UFC1 and Z-HAT may make the protein susceptible to degradation mechanisms in autophagy, which could be a focus for future research. Ultimately, the results give implications that UFC1 could be used as a potential therapeutic to help treat alpha-1 antitrypsin deficiency, but further research on the mechanism of its function is necessary. Additionally, AAT substitution will remain necessary to ensure anti-elastase activity. Thus, it is not a completely efficient therapeutic to treat both effects of the disease-causing variant. This study opens the door for future research of small molecule therapeutics as a treatment option for alpha-1 antitrypsin deficiency.